Hello everyone, my name is Carolina Ugarte. I'm a second year student of the Iracom program and today I will be presenting on the topic of a novel multifunctional edible coating based on sodium alginate and whey protein isolate designed for banana preservation. This novel solution aims to extend the shelf life of bananas. Bananas are a highly consumed fruit, rich in essential nutrients and vital for the economy of many tropical and subtropical regions. However, they ripen rapidly due to their high respiration rate and are prone to oxidative damage and microbial infections, resulting in significant post-harvest losses of 20 to 40%. For this reason, this project aims to develop a water-resistant, self-healing edible coating that can extend the shelf life, protecting them from ruin and decay. The main components of the coating include sodium alginate, known for its film-forming abilities but poor water resistance, whey protein isolate, which enhances mechanical properties and water resistance, and also gluconolactone that is used to acidify the solution gradually, strengthening the interaction between sodium alginate and whey protein isolate. The films are prepared by mixing these ingredients, casting the solution, and drying to create the composite coating. The preparation process of gel films is shown in the diagram. The gluconolactone decomposition was used to produce gluconic acid, which induced the protonation of amino groups on whey protein isolate. The strong electrostatic interaction between the positively charged whey protein isolate and the negatively charged sodium alginate induced solution gelation. For this work, various ratios of sodium alginate and whey protein isolate were used to create the films. Now let's talk about the mechanical properties. This part is important because the excellent mechanical strength and flexibility of the composite coating can prevent the fruit from mechanical damage during storage, transportation, and marketing, and avoid the shortened shelf life caused by mechanical damage of the fruit. The composite films demonstrated significant improvements in several areas. The tensile strength increased by 37.5% with the introduction of the whey protein isolate, while the elongation at break improved, showing enhanced flexibility. In this case, Figure C illustrates the water contact angle of the composite films. As whey protein isolate content increase, sodium alginate decrease. The film become more hydrophobic. For example, the water contact angle of the S10W10 composite was 2.16 times higher than that of pure sodium alginate film. The addition of whey protein isolate also enhanced surface roughness, further boosting hydrophobicity. In Figure D, the pure sodium alginate film dissolves quickly in water, showing poor water resistance. As whey protein isolate content increase, the film swelling rate decrease, indicating improved water resistance. The S12W8 film had a soluble solid loss rate of only 55.1%, demonstrating that whey protein isolate enhanced the water resistance of the sodium alginate film. This is another interesting result. The self-healing capability was notable, as the scratches could be repaired within 20 minutes when hydrated, maintaining over 75% of its original mechanical properties post-repair. Talking about the structural analysis, SEM imaging revealed that the composite films have a uniform, compact structure with increased surface roughness due to the presence of whey protein isolate. This roughness contributes to the improved hydrophobic properties. Additionally, the cross-linked network formed by the interaction between sodium alginate and whey protein isolate enhances for water resistance and mechanical strength of the films. Moving on, the composite films exhibit excellent barrier properties, reducing water vapor, carbon dioxide, and oxygen permeability. This is essential for minimizing water loss and controlling the respiration of fruits, which in turn helps extend their shelf life. Now let's talk about the banana preservation experimental setup. To test the effectiveness of the coating, 120 Cavendish bananas were selected and divided into four groups. The first one is the control with no coating, the second one with a commercial polyethylene coating, the third one the sodium alginate coating, and finally the sodium alginate and whey protein isolate S12W8 coating. All of them were stored at 25 Celsius degrees and 50% relative humidity, and their conditions were monitored over 8 days. The first parameter to analyze is the visual appearance. As we can see in the picture, by day 3, bananas in the control and polyethylene coated groups showed signs of browning, while those coated with sodium alginate and S12W8 remained visually appealing until day 8. These are another important results. The S12W8 coated bananas experienced significantly lower weight loss, only 42.9% of the weight loss seen in the control group. Talking about the firmness, the S12W8 coating effectively preserved the firmness of the bananas. After 8 days, S12W8 coated banana had a firmness of 19.8 newtons, that is more than double the control group. This indicated that the coating delayed the ripening process, maintaining the fruit's texture for a longer period. Analyzing this, the pH and the acidity, we can see that as bananas ripen, their pH increases and acidity decreases. The S12W8 coating maintained a lower pH and a higher acidity compared to the other groups, signifying the late ripening. This helps keep the fruit fresh for longer. The polyphenol oxidase activity, which leads to browning, was lower in S12W8 coated bananas, peaking later than in the control and polyethylene groups. The browning index was also significantly reduced, being 60.1% lower in the S12W8 group compared to the control group, showing better protection against oxidation. In summary, the S12W8 composite coating demonstrated superior preservation performance, delaying ripening, reducing browning, and maintaining firmness. This innovative, water-resistant, self-healing coating holds promise for a broader application in extending the shelf life of perishable fruits and improving sustainable packing solutions. I just want to add that I choose the present paper because it's pioneering topic. And when searching in Web of Science, this paper was marked as a highly cited paper, meaning that it has received enough citations to rank in the top 1% of publication in the field of agricultural sciences. It was published in June of this year, and this research has already gained 9 citations, underscoring its impact. Featured in Food Hydrocolloids and Elsevier Journal with a 2023 impact factor of 11, the study benefits from the journal's strong influence in food science. Finally, I just want to say thank you for your attention and interest in this presentation.